Good morning. I want to thank the organizers for inviting me to speak, and happy to be here today. I'm going to tell you today about a new technology that I've been involved in the development for automated single-cell expression profiling in intact tissue using proximity ligation in situ hybridization. I just want a disclosure. I'm a co-inventor on a Stanford patent for this technology. Per Harbury is a colleague in biochemistry who's the co-inventor, and he and I had kind of put our heads together. He knows everything. He's forgotten more about molecular biochemistry than I've ever known, and I'm a biologist, and we thought, hey, but he's very interested in biology. So we sat about and we thought, well, what would be the ideal kind of single-cell profiling technology? And so I've got a list of features here. It would be great to detect what's expressed, so you don't have to pick certain genes, and you don't have to have prior assumptions. It would be good if you could detect all different kinds of RNAs, not just messenger RNAs, but non-coding RNAs, et cetera. It would be great if it was compatible with detection of other molecules, for instance, proteins, and you could integrate and layer the different levels of expression. Spatial information, we thought, would be very important. And it should be non-cumbersome, so it should either be very rapid or automated to generate omic scale data. And technically simple to perform and analyze, and that you would have no need for access to specialized or expensive equipment. So I'm sorry that I don't have this technology, but I want to talk to you about what we do have and how it's very complementary to single-cell RNA sequencing. So first, I just want to mention there is a lot of effort. A lot of groups are working on different kinds of technologies to combine transcriptomic with spatial data. So there's something called spatial transcriptomics through spatial barcoding. And so there's something called physique and other kinds of technologies that have been published. And in their current iteration, they really can't match any of the needs because they're expensive, they're specialized, they're very low sensitivity, so really all they can do is, because it is sequencing in the tissue, they can detect what's expressed. They're also, unfortunately, not single-cell resolution. The level of resolution that's been achieved is far larger than a single cell. But a lot of people are working on this, and this would be great, of course, because if they could get these other features, it would be wonderful. So single-cell RNA-seq, so we've heard a lot about, and so this is great. It detects what's expressed. As you heard from Kyle and Astrid, you can just throw your cells in there and be surprised at the results that you get because you don't have any upfront biases or assumptions. It's non-cumbersome. You have to just collect your cells and send them for sequencing. But it doesn't give you spatial information. It's expensive, and it pretty much only detects messenger RNAs. So it's got some limitations, although it has been obviously transformative. What I'm going to tell you today, and there are several other technologies, is in situ hybridization. So our technology we call PLISH, and there are many others that have been published, including MERFISH, SeqFISH. It's commercially available through a company called RNAscope, and there's others. And so the good thing about in situ hybridization is that you can detect any kind of RNA you like. You don't have to depend on polyadenylated sequences. You have a fixed tissue, and it's compatible with immunostaining and other kinds of orthogonal approaches. You get spatial information at single-cell resolution. It's technically simple to perform and analyze. You don't need any special equipment. It's not expensive. However, you don't just detect what's there. You have to pick what you want to look for. So that's a big limitation. It's also cumbersome. You can, it's a little bit of a pain to do, and it's not automated. So these are limitations. But if you put it together with single-cell RNA-seq, they're really highly complementary. I think they they really work well together. And I'm going to show you an example today of how we've used this approach. So what does in situ hybridization bring to the table? So these, I think, are really important. So one thing is that unlike immunostaining, so immunostaining is another way to do single-cell analysis in situ, you can do what I like to call on-demand marker selection. You don't need to have a commercially available antibody that works in tissue to look for what you want to look for. And this is a huge limitation of immunostaining. If there's no commercially available antibody, which for most gene products there's not, you're out of business. So this is really important, and this is actually initially we were thinking of multiplexing antibodies, but then we recognized that this would be a big limitation. The other thing is that you get the spatial information and histological correlation. So I think a lot about disease, and my own clinical practice is focused on interstitial pulmonary fibrosis and other interstitial lung diseases. And in these diseases, you've got these very stereotyped lesions that are very recognizable, and that forms the basis of diagnosis. And so, you've got a very there's a lot of signaling going back and forth and interactions that are mediating the disease process that it would be very important to integrate. And if you dissociate the tissue and you can't reconstruct it, that's going to be very hard to do. And the other thing is you can actually even look at the cytological data. So you can look at the cytology of a single cell, and this could be very important for diseases like cancer and looking at precancerous areas where you can correlate the molecular and the cytology. It's also currently more sensitive than single-cell RNA sequencing. So we did some comparisons in our paper where we looked at SMART-seq RNA sequencing that had been published by Steve Quake's lab in HEC cell line. And there was a lot of dropout of even moderately abundant transcripts. So I think SMART-seq2 is more sensitive. I think it's better, but I think this is a big concern is the level of sensitivity. The other thing is that it's been published now, and people are recognizing that when you dissociate a tissue, you induce alterations in gene expression. And so people have actually published the classes of genes that get induced, and so you're not actually sure that what you're looking at is what's going on in the tissue at that time. The other thing is that all cells in the field are analyzed, so you're not going to miss particular cell types that are either very rare or that are just morphologically difficult to capture. Finally, you heard about the rapid response team. One nice thing is you don't need to really be on call for when a tissue is available. As long as you are friends with your surgeons, which we try to be, we send them fruit baskets every year. We can get them to just throw the tissue in formalin, and then we can sleep peacefully. Okay, so this is the technology. I'll just describe it real briefly. We call it proximity ligation, in situ hybridization, or PLISH. So what it involves is you have your target RNA, and you target it with two sequence-specific probes that we call H probes. And so they're sequence-specific to the target, and then they have these overhangs. Now these overhangs, they form kind of a platform on which we can then add on what we call the circle and bridge construct. And so these are nucleotides that bind in a sequence-specific manner to the overhangs, and they contain these red regions indicate bar-coded sequences that are going to be used for detection. So we can make this any sequence we want, and we know that the complementary detection oligonucleotide is going to have a red fluorophore and bind to these sequences. You then perform a ligation so that you generate a closed nucleotide circle, and because of the platform, it has its own free 3' nucleotide end, and you can just do rolling circle amplification. Now, rolling circle all of these things have been around for a long time: proximity ligation, rolling circle amplification, et cetera. But rolling circle amplification is really amazing because it is so incredibly bright, the signal, and you find a punctum, a single really bright punctum. So unexpectedly, we found that it really renders, at least in mouse tissue, it renders autofluorescence trivial, and you can also distinguish a real signal from a non-real signal because it's a discrete punctum of a certain size. Now, what this allows us to do is we can tile along a messenger RNA. So for non-abundant targets, if you just target one part of the messenger RNA, if that part is protein bound or some other reason it's not accessible, it can be hard to detect. And so by tiling, we can really increase the efficiency of our detection. And so then we can do this, and we can barcode 4 or 5 different fluorophores that can be detected with a conventional fluorescence microscope and do a 5-color in situ hybridization. So this is what it looks like in cells, and so this is SOX4 in, I think this is in HEC cells, and you can see these discrete puncta that are the rolling circle products. So in this case, we used one probe pair to target the SOX4 mRNA. Now, if we tile and we use 10 probe sets, we greatly increase the efficiency of detection. And this is very helpful in particular for low abundance targets. And then if we mismatch the probes so that they don't bind next to each other, then you don't get any signal. And so this really is a feature of the proximity ligation, that it ensures very high specificity because you have to have two independent binding events right next to each other in order to get a signal at all. It's compatible with immunohistochemistry, and so here I'm showing you an antibody for surfactant protein C and the in situ for surfactant protein C and the very good co-localization. And as I mentioned, I think it's very important you get the spatial information, histologic and cytologic correlation. So this is human skin, and this is in situ hybridization for keratin 5. And so you can see in the epidermis, you can see the nice basal distribution of these basal cells. This is a human lung stained for surfactant protein C, and so you can see these nice cuboidal alveolar type 2 cells. And this is an example of a patient with idiopathic pulmonary fibrosis. And so this is staining for SPC, and you can notice a lot of things. One is that you can see the epithelial cells that are facing a lumen, that there's a lot of SPC. So there's too many type 2 cells compared to in the normal alveolus. And you can also see SPC expression in cells that don't appear to be epithelial. We don't know if they started out as epithelial, but they're not facing a lumen currently, and they're expressing lower levels of surfactant protein C. So this histological correlation is very helpful because if we dissociated this tissue and we saw all of this variability, we wouldn't know really where the cells were, and we wouldn't be able to see this pattern. The other thing I want to show you is the cytological information. So you can see in the normal lung, you can see these cuboidal type 2 cells, which are normal, but then in the pulmonary fibrosis, you can see this incredibly flattened squamous cell that is SPC positive. And so again, this is information that we would miss if we didn't have it in situ. Okay, I'm going to show you now an example where we combined in situ with single-cell RNA sequencing to really make some novel insights into lung biology. So I'm going to talk to you about Wnt signaling, which is a large family of short-range secreted ligands. And it binds to frizzled, and by stabilizing beta-catenin, it acts as a transcription factor. And really importantly is there are known transcriptional target genes of Wnt. So we love this. So we can actually stain for axin 2 or LGR5, and we can identify cells in which Wnt signaling is active. And we knew that Wnt signaling has been reported to be active in adult stem cells in many different tissues. So we were interested in the idea that could Wnt signaling be regulating type 2 stem cells in the lung? So we did an in situ hybridization for axin 2 to ask whether axin 2 is expressed in type 2 cells. And if you do that, you can see here, I'm going to show you an example here, that about 90% of the type 2 cells, shown here by SPC, did not express the Wnt target gene axin 2, but that 10% they did. You can see these puncta for axin 2, and this was reproduced through multiple different animals. And so this was exciting because it suggested that a small subset of the type 2 cells were undergoing active Wnt signaling. And so then we used the Cree-ER system to label alveolar type 2 cells that express Wnt by using the axin 2 Cree-ER. So these are the ones that are receiving the Wnt, rather, that are Wnt active. And we followed them over time, and as you can see here, this is a multicolor approach, similar to what Astrid was showing you, that you can see a single axin 2 positive type 2 cell, and you can see it expands into a clone of multiple type 2 cells. This is SPC staining here. And so if you quantify that, you can see that over 6 months, the majority of the Wnt active type 2 cells proliferate. And this is much more than the Wnt inactive type 2 cells, because they sort of take over the population. Okay, so now we know that Wnt is a very short-range signaling factor. And so we want to know, well, okay, these type 2 cells are receiving the Wnt. Where is the Wnt coming from, and what Wnt is it? So this is single-cell RNA sequencing data where we looked at a large number of type 2 cells. And it turns out that type 2 cells themselves don't express Wnt. So it's not an autocrine mechanism in this case. But we knew that from classical literature that Wnt is that fibroblasts in type 2 cells have a very close relationship, and in spheroid assays, fibroblasts help type 2 cell proliferation, so they were a good candidate. So we looked at fibroblasts, and we found that indeed a significant minority expressed Wnt5a. So now we said, okay, if this is the operative Wnt, and if this model is correct, then if we stain for Wnt5a, axin 2, SPC, the fibroblast marker, we should be able to identify these signaling centers. And so that's what we did. And so you can see here that your conventional alveolar type 2 cell that is axin 2 negative is not undergoing Wnt signaling. It's not near a source of Wnt. But when we found these axin 2 positive type 2 cells, we found them right next to these Wnt5a expressing fibroblasts. So it was really beautiful. We could quantify this and show that it was highly enriched. And we were able to then use strategies to target and block Wnt secretion from fibroblasts and show a reduction in the number of the axin 2 positive type 2 stem cells. So this was, we couldn't have done this because there aren't antibodies for Wnt, and so it was really a nice synergy between single-cell RNA-seq and the in situ method. Okay, the other thing that I'm going to tell you about is that we developed a way also to do iterative staining. So you can easily erase and detect a large number of transcripts. And basically how it works is you would do your hybridization all at once and generate all of your rolling circle products, and then you would just label 4, image, erase, label the next 4, and you could iteratively do this. And once you're done, you have a bunch of images, and you can use Cell Profiler to do automated single-cell quantitation, and you can generate heat maps and t-SNEs and cluster and do all kinds of downstream analysis, and then you can also remap onto the tissue the different cell populations of interest. So this is basically how it works: is you do your initial hybridization, you detect a subset, and then you erase them, you detect the next subset. So you can still do this with a conventional fluorescence microscope. You don't need any special equipment or anything like that. So we did that, and this is iterative push. We used 4 cell type markers and 4 housekeeping genes in mouse lung, and so we would stain for 4, as well as DAPI, take a transmitted light image, erase, and then detect the next 4. And when we did that, we then used unsupervised clustering. We didn't give any information to the software, and it was able to reproduce all of the cell types that we were staining for, and you can see them displayed here in a t-SNE. And so this was about 3,000 cells from several different fields of view that was analyzed in this automated manner. We then spatially remapped the cell classes. We said make the club cells cyan, make the type 1 cells red, type 2 cells green, and as you can see, really beautifully, the club cells are all where they should be, and type 2 and type 1 cells are where they should be. So this again affirmed that it was working accurately. Now, I want to just point out one thing, too, is that we can actually remap individual cells because it records the spatial information. So there are these cells that have been described that are expressing both a type 2 marker and a club cell marker at the bronchoalveolar junction. So we saw them on the t-SNE, and we said, OK, computer, tell us where is this cell? And it's right there. So it's exactly where you'd expect it to be. So we can even pull out these rare cells that we can detect. So it's a very powerful technology. And we can also reveal new biology by looking at the spatial information. We notice here that we found two subtypes of a club cell, and that's not been described before. And really the thing that distinguished them was beta-actin. There was high level of actin expression in one and low in the other. And so we said, geez, is this a real thing? You can see it here with the actin heat map. And so we said, OK, let's remap these. Let's color them 2 different colors and remap them. And what we found was very striking is that you can see the actin high cells here in the lighter blue are very highly enriched at the very ends of the bronchioles, whereas the actin low are more internal. So this is probably not a different cell type or anything, but I think it does reveal something about the biology that we would not have been able to understand without spatially localizing them. OK, just in the last few minutes, this is a collaboration with Edda Spiekerkatter. So like the Wnt signaling family, BMPR2, you guys have probably heard of BMPR2, I'm sure. Secreted ligands, and it's a receptor. This is how it works. And like Wnt signaling, there is a transcriptional target, and maybe more than one, which is ID1. So we're very excited that, hey, we can use our in situ to look at human lung and sort of interrogate the BMPR2 signaling activity. And of course, as you know, more than 75% of hereditary and 25% of sporadic PH involves BMPR2 heterozygous mutations. So this is the project now of Adam Andruska, who's at the ATS, so he can't be here today, one of the pulmonary fellows. And so he wants to use PLISH to generate a molecular atlas of BMPR2 signaling. So the idea is to localize BMPR2 by in situ hybridization along with a panel of markers, including cell type markers, so you can identify the different cell types, EMT markers, proliferation markers, other markers, and do this in conjunction with looking at signaling activity, so staining for ID1 and also combining this with proximity ligation to look at for phospho-SMAD activity. And the tissues that we plan to use are non-PH lung, known BMPR2 mutant PH lung, idiopathic PH, where we know there's not a known TGF-beta pathway mutation, and connective tissue disease-associated PH. These are the tissues that we can have access to. And then this can be, once we have this basic map, we can refine it by looking at different stages of disease in the same individual and try to learn something about the dynamic progression of pH. We can integrate it by co-staining with validated antibodies and expand it by looking at additional signaling pathways. So I think it's going to, it's something that can really be built upon. Now, we haven't made as much progress as we had hoped. I think that's usually the case for my lab. But I'm going to show you just what it looks like here. So this is, first, Adam went to validate the probes, which is hard to do in humans because a lot of genes we might want to look at, we don't know the expression pattern. So this is, he used silencing for BMPR2 and stained for VE-cadherin and BMPR2 in pulmonary artery and endothelial cell line. And so you can see here in green is the VE-cadherin and red is the BMPR2. And so this is the knockdown of BMPR2 and this is the control. So and then when he quantified it by Western blot and then also by using cell profiler, you can see that it seems to be, it worked well at selectively knocking down BMPR2, and then he was detecting less signal. This is in non-PH lung. This is our normal lung, which is actually, from a cancer patient, resected portion that's not involved. And this is staining for VE-cadherin, smooth methyl actin, BMPR2, and DAPI. Now, immediately you're going to see what you all know is this highly autofluorescent elastin. So Adam came up with a clever way to erase the autofluorescence, which I don't have time to get into. And so now you can see you're left with BMPR2 here in red, smooth muscle actin in magenta, and VE-cadherin in green. And just to give you like a closer up, here's the lumen. This is the VE-cadherin, smooth muscle actin, and this is an alveolar sac, I believe. And so when Adam used, we want to do this in an unbiased way, so when he went to quantify the signal using cell profiler, which first identifies the nucleus and then measures the signal around each nucleus, this is what he found. So on the left here are alpha SMA high, VE-cadherin low smooth muscle cells, and they have low levels of BMPR2. And over here is a alpha SMA negative VE-cadherin high, which has higher levels of BMPR2 expression. So we're not ready to hang our hat on this result, but this is the kind of way that we want to try to analyze it as we can improve the quality of the stain in the human tissue, which are all sort of ongoing projects. So just to summarize, so I think multiplex in situ approaches, either PLISH or another approach, which are going to become more and more widely available. And it's an ideal complement to move forward from these single cell data sets that are emerging, like the cell atlases. You can pick, because you can multiplex, you can take an entire signaling pathway of interest, and you can focus on that, and you can reveal dynamic signaling interactions in situ that may be involved in disease, and you can try to then confirm them using phospho antibody staining. And it provides a combined molecular cytologic profile of the cells. You can correlate them with disease stage. We have a similar project in pulmonary fibrosis where in any given individual, you've got all different stages of disease. So it's a perfect situation where we can look at normal lung, an early IPF lesion, middle, and late, and we can try to reconstruct the disease process and understand what are the dysregulation that is occurring early in the disease or even before the disease manifests versus the late effects. And you can use this to generate a comprehensive single-cell molecular atlas with the spatial and molecular information. So these are the people that did the work. This is Per, my collaborator in biochemistry. Monica and Dan Riordan worked on developing the PLISH. Ahmad is a graduate student who did the WNT work, and then Etta and Adam involved in the pulmonary hypertension aspect. Doug Brownfield is a postdoc who did some of the RNA-seq analysis. And then I want to thank Marlene and Mark Nichols for useful conversations and our funding source. So thank you very much. Sorry I went a little over.
Mark.
Really nice, Tushar. Have you encountered the problem of changes in cell identity with disease? So you can construct nice maps in health, and then to the extent that cells may change identity, you're not looking at surface expression so much as cytosolic expression of things. So have you encountered that?
Yeah, so right now we're sort of, because validating the specificity of the staining turns out to be, as we look for things that have never been looked at, it turns out to be quite laborious. We're trying to do different approaches like knocking it down in cell lines and then showing that the signal goes away and things like that. So we haven't done too much stuff in disease, but I think that's going to be a big challenge. And I think we've seen this. There have been a couple of single-cell RNA sequencing data sets published on pulmonary fibrosis. And you look at it and it's just, there's so many different types of cells, so many different molecular profiles. It doesn't make sense. So I think it's going to be a big challenge. I think just disease is just going to be, going to have a spectrum of molecular phenotypes compared to healthy tissue. So I don't know what, I don't know an easy way to figure it out, but I think people are going to have to just kind of cluster, see if they see the same kinds of cell types in different individuals and try to understand what they're doing.
Yes. Mikaela.
Thank you. Very elegant work. So I had two questions. The first is a very simple one. Do you need to do anything special to preserve the RNA, or can this be used in any formalin-fixed tissue that people have sitting around in the lab?
Yeah, that's a great question. So we have found that we kind of went around the world. We first tried paraformaldehyde, and then we tried all these different things ranging from something called Mu glue to methanol, and we've ended up back at paraformaldehyde. So formalin is quite good. The key thing seems to be putting it in formalin right away. So
Sure.
We've tried old blocks, really, really archival old formalin fixed paraffin embedded, and it can work for high abundance genes, but I think you do take a hit, especially if you don't know how it was collected. If you just have an old block, you don't know how long someone waited before they put it in the fix. That's a challenge.
Okay, thank you. And then the second question is the Plink method sensitive enough that if you wanted to do dual detection of a DNA rearrangement and then also gene expression, for instance, in a lung cancer, it's challenging to do the in situ hybridization on the single copy DNA rearrangement. So I wondered whether, can you combine these two together? Yeah,
that's a great question. You've been spying on our lab meetings. So we're very interested in KRAS, G12D, and lung cancer, and so we've been working very hard to try to detect the RNA transcript of that, and we think we can do it. We're not, but we need to really validate it. But we think we're seeing real signal, and the way that works is just to do a proximity ligation directly on that point mutation. And so you can't tile, so you take a hit in sensitivity, and you can't pick and choose your target gene. So I think it's challenging, but it looks like it's working the way that we have it for KRAS G12D. DNA is harder because two copies unless you've got amplification. So we haven't tried that, but I think RNAscope is trying to get staining for that. So I think hopefully someone will make a breakthrough and it'll become more doable.
Thank you very much.